Greetings everyone, my name is Shannon Rodrigues and we are going to discuss some aspects about the organogenesis of the visual system. The visual system is an important component of the central nervous system through which we perceive our surroundings. The visual system includes the eyes and the connecting pathways of the central nervous system to the visual cortex and other parts of the brain. The visual system has an immense contribution to the survival of the human race to this day and age. Hence, let us see as to how this amazing organ comes to be. The organogenesis of the eye can be mainly divided into four important factors, and each factor with its characteristic. So the first factor is the dynamics of the eye development, and its characteristic features are the involuting ectoderm and mesoderm interaction with the head ectoderm to form a lens-forming bias, a precise spatial relationship of the lens and retina, and the lens placode formation due to optic vesicle and diencephalon. So let's look at what each of these means. The major sensory organs of the head develop from interactions of the neural tube with a series of epidermal thickenings called the cranial ectodermal placodes. As you can see in image B, you can see the lens placode, which is responsible for the development of the human eye. So around week 3 of gestation, that is day 24 or 26, we can see the appearance of an optic groove in the diencephalon area. Again, you can refer to image B. As you can see, an evagination from the optic groove, which is the optic vesicle, will move towards the lens placode. This movement is induced by the surrounding mesenchymal cells present. So, this optic vesicle, on contact with the lens placode, will become the optic stalk, which we know as the optic nerve after further development. So the lens placode, as you can see over here, it invaginates towards the optic vesicle. Okay, so the edges of the lens vesicle, they pinch off, and then the lens is present in the optic cup. The single layer optic cup has now become double walled, and the lens is present in it. So along the ventral surface, we get the presence of the hyaloid veins and artery. As the optic cup fuses, it leads to the entrapment of the hyaloid vesicles in the optic nerve. This hyaloid vesicle along the ventral surface becomes the retinal artery and vein. The distal hyaloid vessels, which supply the developing lens, degenerate into a vitreous body, leaving a hyaloid canal. So along the ventral surface, we get the presence of the hyaloid veins and artery, which will supply blood and other nutrients to the developing eye. Now, let us take a look at the various transcription factors responsible for the organogenesis of the human eye. The first is 6-3, which is responsible for the proliferation of retinal precursor cells. PAX6, sonic hedgehog, and PAX2 are play a pivotal role in the development and organogenesis of the human eye. The organogenesis of the human eye is characterized by the separation of the single eye field into two different eye fields, and this is dependent on the secretion of PAX6, one of its family members called PAX2, and the sonic hedgehog, without which we would have a condition known as cyclopia, that is only one eye, that too at the forehead. RX1 plays an important role in the spatial patterning and the morphogenesis of the optic vesicle. So later stages, it is localized to the neural retina where it may be expressed for retinal differentiation and proliferation. On comparison of image A and B, where image A is the wild type, we can observe proper development of the human visual system, whereas in image B, due to mutations in the sonic hedgehog genes, we can see the development of cyclopia. There is no division of the single field into two different fields. Now, let us take a look at the third factor important for the organogenesis of the human eye, which is the neural retinal development, characterized by the development of the different neuronal cell types, which include the photoreceptors, the rods and cones, cell bodies of ganglion cells, bipolar neurons, responsible for transmission of the electrical signals to the brain, the Mueller glial cells to maintain the integrity, Amarkine cells and horizontal cells to transmit impulses in the plane of the retina. The optic cup can be divided into two layers. That is the outer retinal layer, which due to the presence of melanin will give rise to the colored pigmented retina part. And then there is the inner layer, which will further be divided into an outer layer and the inner layer. It was studied that a single neuroblast precursor cell from the germinal layer can give rise to at least three types of neurons, or two types of neurons and one glial cells. Scientists determined this by injecting a virus into the retinas of newborn rats whose retinas were still developing. The virus contained beta-galactosidase gene, which are not present in rat retina, but it could be expressed later. A month after the rat's eyes were infected, they were removed and stained for the presence of beta-galactosidase. So that means the progeny cells would have been stained blue. From image D, we can observe that light first comes in contact with the various nerve layers and then towards the inner rods and cones of photoreceptors. So this gives us an idea as to how the inner retina actually develops. The first to come are the rods and cones of photoreceptors, which are at the innermost side. Then the cell bodies of these very photoreceptors. We have an outer plexiform layer where synapses occur. Then the bipolar nerve layer which carries the synapses forward. An inner plexiform layer again where synapses occurs. And the ganglion cells which will further carry the signals towards the occipital lobe. Now we have arrived at the last aspect of the organogenesis of the eye, which is the lens and cornea differentiation. So we know that the lens pinches off from the surface ectoderm and situates itself now in the double-walled optic cup. So the lens vesicle will induce the cornea formation in the ectoderm. An intraocular pressure is important for proper lens formation. Crystallins are transparent lens-specific proteins. So these are necessary for the differentiation of the lens and its functions. Under the influence of the neural retina, the inner portion of the cell vesicles elongate and differentiates. With, so the lens has three regions: an anterior region of dividing epithelium cells, an equatorial zone of cellular elongation, and posterior and central zone of crystalline forming fibers. So let's take a look at what this actually is. So, during its development into lens, the lens placoid will round up and contacts the overlying ectoderm. So the lens vesicle induces the ectoderm to form the transparent cornea. So physical parameters such as intraocular pressure become necessary for the correct curvature of the cornea, so the light can be properly focused on the retina. A ring of scleral bones will be derived from the neural crest cells, and these are inelastic, which help maintain the intraocular pressure. So crystalline is a lens-specific proteins. Okay, so cells at the inner layer of the lens vesicle elongate, and under the influence of retinal cell will become lens fibers. As you can see, the lens fibers. So the lens fibers, these synthesize the crystallins, which will eventually fill up the cells and will cause the extrusion of the nuclei. As you can see, the nuclei are moving. So crystalline synthesizing fibers continue to grow and fills the space between the two layers of the lens vesicle. Anterior cells of the lens vesicle constitute a germinal epithelium layer from which the lens fibers grow. So we have an anterior zone of dividing epithelium, an equatorial zone of cellular elongation, and a posterior and central zone of crystalline containing fiber cells.